really happy that we have Paul today with us, and it's the first ever talk of the PSH. And so I'm quite happy that we gained you as a first speaker. Okay, I have to raise the expectation. But let me quickly introduce Paul. I mean, we know each other already quite over a decade now. So Paul studied in the UK in Norwich, and over his time in Norwich, he also worked in Bremen at the Max Planck Institute. And thereafter, he did something which is quite clever. He went to the United States, to Berkeley. And then after like 4 years there, the Luxembourg government brought him back with a program which is called the PrEP. What they do is they gain back their talents that they have trained in the United States. And the circle closes after he became full professor for systems ecology. He also went again back to the United States, actually last year or this year, as a visiting professor in Stanford. So the other side of the bay, I have to say the better side of the bay.
Weather-wise, yeah.
Where he stayed for a year and enjoyed the cool science that you have in there. Yeah, Paul, what else do I have to say? ERC granty, ERC consolidator grant. It's one of the prominent grants that you can get in Europe at all. Like close to 200 citations, highest ranked journals, highly cited researcher. So everything top notch, can't be better. So that's why we are now looking forward to your.
All right, how to build up expectations. Thanks so much, Andreas, for that very kind introduction. It's great to be back here at HIPS. I was here last year for your symposium, and I gave a talk there. And for those of you that were there and made it today, you won't be disappointed because I will give a completely different talk, okay? So I will not talk about our work on, for example, antimicrobial resistance and One Health. I will really talk about the human microbiome and its impact in human health and disease. All right, so my pointer is now also working. So we take a microbial systems ecology approach. What we understand under this is that we aim at generating systematic metagenomic, metatranscriptomic, metaproteomic, and metabolomic data. But data generation is, of course, one thing. Much more important is, in our assessment, the data analysis that you do subsequent to this. And so if you sample microbial communities over space and time, and I'll give you some concrete examples about this, we are now able to get to the point of reconstructing the constituent networks of individual populations. And what is more exciting about that even is the fact that we can look at emergent properties. So, for example, microbial molecules that would be the result of interactions that you would not be able to find, for example, if you're studying a given organism in isolation. And in particular, with respect to the impact on the human host, some of those emergent properties may actually be the ones that trigger disease processes in susceptible cell populations. So, for example, I'll give you some insights into our work on Parkinson's disease, where obviously neuronal cells are affected. In other disease, potentially other susceptible cell populations are affected. And so what we're very much interested in is to leverage this integrated multi-omic data to get to the point of unraveling the molecular mechanisms. And the molecular mechanisms may actually involve different molecular entities or different moieties. So they may involve nucleic acids, they may involve proteins, peptides, or indeed small molecules. And this cocktail of biomolecules is what we recently termed the expobiome. So the expobiome is, in essence, the cocktail of biomolecules that is the result of the interactions between different microbial populations, and that themselves may be a trigger or may exacerbate disease processes. All right, so this is a bit of a scheme, right? And to just give you an idea of that, this is in essence not fanciful thinking. Just take one step out of research on the human microbiome and focus on some of the work that we've done on environmental systems. So we've worked, for example, on biological wastewater treatment plants. And I also gave last year in my talk, I gave you some insights into the work that we've done, for example, on glacier-fed streams. So we still work on environmental systems. And some of these systems are really good model systems for the development of the approaches, such as, for example, biological wastewater treatment plants. where we have, through the generation of the systematic multi-omic data, we've been able to gain, for example, insights into the niche ecology of constituent populations. We've been able to identify keystone genes. So those are genes that, in terms of the overall metabolism, have a disproportionate effect on overall ecosystem function. And those are typically encoded by what in classical macroecology we would call keystone species. We've been able to look at what are the mechanisms governing population resistance and resilience. And then, for example, by being able to track the CRISPR complement, so the spacer complement, along with the resident phages, plasmids, and other mobile genetic elements, we've been able to, through the longitudinal data that we've had, we've been able to develop a first model of the community, and that we have built on subsequently that I'll touch upon. And very important now, the same approaches are, of course, pertinent and applicable to the human microbiome. And we still, at this moment in time, still have a clear focus on the gut, although we are also very much interested in the oral cavity, nasal cavity, and other body habitats. But I've already now given you some notion that we're using the high-resolution longitudinal data to develop models. And then these are now actually getting to the point of being quite powerful. This is from a recent paper from earlier this year, where we basically used 1.5 years worth of weekly time data from the local biological wastewater treatment plant at Schifflange in Luxembourg. And then we were from these multi-omic data sets, again, comprised of metagenomic, metatranscriptomic, metaproteomic, and metabolomic data. We were able to then extract the time-dependent information. We summarized this into 17 distinct temporal signals, and these explained the vast majority of the temporal variants. And then we were able to use this 1.5 year worth of data as a training set to then do prediction or forecasting. And here you see the fit of the models in the subsequent years, over 5 years into the future. And this works actually remarkably well. So we had here a nearly perfect trend prediction for the subsequent 3 years. We have a coefficient of determination of 0.87, which is quite remarkable, I think, if you think about the fact that we're dealing with a very complex ecological system, but that it's in some way, obviously, behaving according to some rules. And this is really, I think, remarkable, because it means that, similar to what we're doing with the biological wastewater treatment plant, where we're looking at, for example, the influent, what is the composition of the influent, that would be very similar to looking at the composition of diet, right, in the context of humans. And so if we're able to build such really nice models to forecast, we should be able to do very similar things on human. And so that is indeed what we're aiming to do. Right, okay, so that was enough about the overall scheme. I just wanted to give you a clear example of this. Is not something that has come from out of the blue, but this is really rooted in microbial ecology as we've applied it to environmental systems now over quite a number of years. Okay, so for those of you that have not really gained all too much insight into the human microbiome, in particular the gut microbiome, just a few highlights here, the way we think about this in particular. Others think, of course, along similar lines, but others, again, especially in relation to taxa, see this quite different. So I think what is important is that the gut microbiome is just generally highly diverse. People talk about 1,000 to 1,500 species. I really don't care about that at all, because that is not the functional units that really matter. The functional units that really matter are strains. And if we look at the human microbiome at each individual, we're talking about hundreds of thousands of strains. And it's that level of resolution that we need to get to. I think it's important also in the context of infection biology. Some of you work in infection biology. We know that in infection biology, the level of taxonomic resolution that really matters is the strain level. So to think that it would be different in the context of the microbiome, I think is quite naive. So I think the functional unit is the strain, and we need to be able to resolve our data up to the strain level. It's also clear that these strains then they assemble to form these complex ecological networks that I've highlighted, and those in turn confer emergent properties that themselves fulfill important functions in relation to the host. And if there is a perturbation of those functions, those might be the triggers that would lead us down the trajectory of a disease. And then finally, the other important aspect to consider here is that the microbiome as a whole, especially in our countries, has been dramatically impacted by the Anthropocene. In particular, massive changes in diet, along with, of course, exposure to antimicrobials. And so to bring this into a bit of a different context yet again, in relation to the expobiome, I mentioned earlier that basically the expobiome is comprised of cocktails of biomolecules. And what we have seen, especially over the last decade and a half, once we had applied high-resolution molecular methods to settings of case-controlled studies, what we've seen are apparent imbalances in particular in the gut, in the microbial ecology, as seen here on the top, right? So we have changes in the overall ecology. And at the same time, we see imbalances on the side of the immune system. In particular, in all chronic diseases, we see that as chronic inflammation. And so the question now is, of course, is the spectrum of biomolecules that might be different as a consequence of a shift in the ecology, may that spectrum then be indeed the driver of these chronic diseases, of which the incidence is, of course, growing. So we know now, again, through the work of a decade and a half, and there are, of course, differences from one study to the other, but I think what is safe to say is that there are just changes in the ecology. We can call this dysbiosis, or we can call this a disequilibrium in the ecology, whatever we want to call it, but I think this shift is quite apparent. Another thing that is a common denominator other than those shifts is, of course, chronic inflammation. And so I think it's not too far-fetched to think that one thing would have something to do with the other. But the point is that this is, of course, complex, and cause and consequence are not clearly delineated at present. And that's why we do think, given the complexity of the systems that we're dealing with, but also the methods available also on the bioinformatic front, we think that this kind of microbial systems ecology approach is the one that's really called for. And so over the last decade, we have built up a suite of methods ranging from the wet lab to the dry lab to do exactly this. So to basically take samples, if it's from the environment or from the host, human host in that case, and then we basically extract all of the biomolecules from the very same sample, when our samples are typically snap frozen. So from the extracted DNA, RNA, proteins, peptides, and small molecules, these are all obtained from the very same sample. And why is that important? That is important because what we want to subsequently do is, of course, to coherently integrate this data along with, for example, metadata. Metadata would be, for example, environmental data that we also collect, for example, from the wastewater treatment plant. It would be information on diet, et cetera, et cetera. And so, yes, we are there. We are able to do this. And that sets the scene that we can now investigate causal relationships between shifts in the microbiome and then disease processes. And so here's just a snapshot of some of the tools that we've developed in order to do this. I'm not going to go into detail here. A lot of you will know VisBin, that you have also then expanded on and contributed to over the years. One other tool that might be of interest here, that's now in the second version we will be published quite soon, and that's PathoFact. So this is for the prediction of antimicrobial resistance genes, but also virulence factors, toxins, and then also more recently biosynthetic gene clusters. Okay, so what have we learned so far by applying this whole suite of methods? Because developing methods is one thing. Of course, what we're aiming to do is science. And so the first disease that we applied these methods to was type 1 diabetes. And the major findings from this were, I think, twofold. The first was that we didn't actually see any changes in taxonomic structure between healthy and diseased individuals. And this was a multiplex family study. So we had, from the same family, we had individuals that had type 1 diabetes and then others that were healthy. So what we did see, nevertheless, were that there were differences in the expression of specific functional genes. And very clearly, these genes are encoded and expressed by distinct populations in distinct individuals. So you don't have a given taxon which fulfills always the same function and is differentially expressed in the context of disease, but there is an individual dimension to this, and along with the individual dimension, there's also a family dimension. And then the second point was that the changes in expression were linked to the levels of endogenous factors, in particular exocrine pancreatic enzymes, where we saw that there was a marked reduction in terms of the levels in type 1 diabetics. And then more recently, what we found is actually that a lot of those changes in terms of taxa conferring functions that are differentially expressed, you already see those in the oral cavity. And that means that generally, across at least that disease, according to our results, basically there are shifts in ecosystem services. So those are the functions that are conferred by the microbiome to the host. And so I'm not going to dwell too much on type 1 diabetes here. I'll talk primarily about our work on Parkinson's, but before I do that, one other aspect to complicate things a little bit is the fact that I highlighted earlier that a lot of biomolecules are involved, right? So cocktails of distinct biomolecules. And so these biomolecules that interface with us as the host, they are, for the most part, the relevant ones are the ones that are extracellular to the microbiome. So that basically would be, for example, either actively or passively secreted by the microbiome. And so in order then to expand on the toolbox that we've built, we've also now built the ability to take from a given microbiome sample, such as, for example, a fecal sample, we can also extract the extracellular DNA, extracellular RNA, extracellular proteins, and extracellular metabolites, again, from the same very sample. And we can also, at the same time, extract the intracellular biomolecules. So that means that we can then do systematic comparisons between intracellular, extracellular, but also, more importantly, compare whether individuals with a given disease would have a difference in terms of the spectrum of extracellular biomolecules that they would be exposed to in the gut, because, as I alluded to earlier, that spectrum of biomolecules that might be imbalanced may be the driver or the trigger for disease processes. Okay, and so the take-home message here is that what you see is both at the level of taxa and functions is that if we look at, and it was for 4 healthy individuals, this is just the methods paper, or data from the methods paper, and what you see is basically at the level of taxa, if we look at intracellular DNA for those 4 individuals, that would be what you would typically resolve in a metagenomic experiment, right? Where you would extract DNA and you would sequence that in a shotgun manner. And so what you can see is that based on the intracellular DNA, you can even not directly extrapolate to the extracellular DNA fraction. You cannot extrapolate to the extracellular large RNA fraction, and so on and so forth. So what that means is if you're doing metagenomics, that doesn't necessarily tell you what is the biomolecular spectrum that you as a host are exposed to, and doesn't tell you how that may be shifted or changed in the context of the disease, and doesn't get you onto the route of actually identifying molecules that would be relevant, not least in relation to disease causation. And why does this matter? Well, it just matters based on, for example, again, these 4 healthy individuals, we were able to look at the extracellular biomolecular moieties and see where there were specific enrichments. And what we found, for example, is that in both the extracellular DNA and extracellular large RNA, there was a clear enrichment in CpG motifs. In the extracellular large RNA fraction, we saw that specific taxas show clear enrichments. So in particular for Faecalibacterium genus, we found that 22% of the reads across all of the individuals were mapped to that particular taxon. And then for one individual, we found up to 5% of the extracellular large RNA reads were derived from Methanobrevibacter smithii, although that taxon is typically, in a metagenomic analysis, less than 1%. For the extracellular small RNA, we saw enrichments in known suspects such as tRNA, rRNAs, and non-coding RNAs, but these were primarily encoded by members of the Clostridiales. And then the extracellular protein fraction was enriched in membrane vesicle associated proteins, mostly from the Bacteroidales. And then finally, in the extracellular metabolome, we find molecules that you would expect to find, but also that have an immunomodulatory function. So combined together, this, I think, shows the fact that we need to look at all of the different levels in order to understand how shifts in ecology may, of course, then impact the host, because it's not going to be a single molecular group that will be involved. Okay, so that is what we have done on the method development front, going from the wet lab to in silico analysis, but also what is important is that you want to integrate these findings with existing knowledge. And so in order to bring together the existing knowledge, we've developed the expobiome map. And I know that this is not readable, and that is for a very simple reason, is that this is already quite complex. So this map is in essence the result of having mined 36,000 individual peer-reviewed publications. And what you have are basically taxa here on the left. Then you have molecules that will be produced by those taxa, human pathways, in particular immune pathways that will be triggered by those molecules, and then how those immune pathways may be in particular dysregulated in the context of a range of different diseases. And this knowledge graph brings together, in essence, the existing knowledge. And what is important about it is that we can then map our own data on top of this and highlight areas that you might expect based on the prior art to be affected, but also find gaps of where we would find, for example, molecules that would be differentially abundant in the case of certain diseases, and try to figure out how those then may indeed impact host physiology, not least in relation to triggering or exacerbating disease processes. And so the challenge here is rather significant. So based on our initial analyses, we know that there are millions and millions of distinct biomolecules that will be involved. And the vast majority at the moment of those are, of course, unknown with respect to their identity, and they're also unknown with respect to their immunogenicity, and of course other processes that they may be involved in. And so an area that we're currently actively working on, and I know that you have also the same interests here, is how those molecules, for example, metabolites here, or other basically microbiome-derived molecules such as amyloid proteins, how they may indeed trigger processes leading to neurodegeneration. And there are two main routes whereby microbial molecules may trigger a neurodegeneration. The first is via the vagus nerve, where you, for example, could have misfolding and aggregation of, in the context of Parkinson's disease, alpha-synuclein, and then you get propagation of the misfolded protein along the vagus nerve. And indeed, from earlier results, we know that the brain stem in Parkinson's disease is affected earlier or earliest before typically the motor symptoms occur. And the second route is, of course, via the circulatory system. And so what one sees is that there are certain commonalities between different neurological conditions, including stroke, Alzheimer's disease, and Parkinson's disease. And in all of these contexts, shifts also in the microbiome have been established. The question now is, of course, is this again cause or is it consequence? Okay, so just a few words on Parkinson's. So this was first described by James Parkinson in 1817. It's characterized, of course, by the classical motor symptoms, but also important to highlight that preceding these motor symptoms, there are a number of other symptoms that have a clear link also to the gut, for example. And in that, many Parkinson's disease patients, typically 60 to 70%, are actually constipated. So there is already an indication that the gut microbiome or the gut as a whole may be somehow involved. And what we do see is that there's a growing incidence, of course, in this disease. So other links to the gut microbiome in the context of Parkinson's disease is that the truncal vagotomy, which is typically performed, for example, for heart arrhythmia, that actually decreases the disease risk. And then about a decade ago, in particular, researchers in Nantes in France started seeing that enteric neurons, they exhibit actually Lewy body inclusions very early on. Again, in individuals that have not had a diagnosis yet for Parkinson's disease, because they do not have the classical motor symptoms. And also now way over a decade ago, Heiko Brack, at the time very famous German neuropathologist, had basically come up with this two-hit hypothesis whereby either Parkinson would be caused by a so far unknown pathogenic agent that would enter through the nasal cavity and or the gut. And so again, how this would look from a mechanism point of view is that you would have a trigger, for example, in the context of the gut. You would get misfolding, for example, an aggregation of alpha-synuclein, the spreading of this via or across the Meissner and Auerbach plexi, along the vagus nerve, and only at relatively late stages then is the dorsal motor nucleus of the vagus nerve affected in the brain stem. And it's from there out that you would then get the spreading. And only at late stages of the disease is then the cortex affected. So we did some work very early on, along with a Finnish group on this, whether there were indeed differences in the gut microbiome in Parkinson's disease. And what we found was that not common signatures to all Parkinson's disease patients, as you see here, but that there is a spectrum of taxa that occur nevertheless at a higher incidence in Parkinson's disease patients compared to healthy individuals. And in particular, what we had seen was this enrichment in Akkermansia. Again, not in all Parkinson's disease patients, but in a non-significant number. And what is also interesting from the functional point of view that I highlighted earlier, so Akkermansia, the type strain is Akkermansia muciniphilia, and as the name suggests, it likes mucus. There are other taxa in the gut microbiome that like mucus, that like foraging on mucus, in particular Bacteroides. And what we see actually, if there is not a high level of Akkermansia, you have the same niche being filled by another taxon, such as, for example, Bacteroides. And I'll come back to this in a few moments' time. But this signature of this increase in Akkermansia has now been, validated across very many studies internationally. And so another aspect to this is that Parkinson's disease is not a single condition. It may actually have a number of comorbidities. And one of the other symptoms that may co-occur with Parkinson's disease is mild cognitive impairment. So a lack in motivation. Recently, there have been a paper from a Chinese group highlighting differences within the context of Parkinson's disease between individuals that had mild cognitive impairment and those that did not have mild cognitive impairment. We were quite interested to see whether our early signatures also showed potential differences. I highlighted now the fact that Bacteroidetes might, for example, occupy the same niche as Akkermansia, and so we were interested to see whether there would be a signature there also in relation to mild cognitive impairment. But what we see is absolutely no signal whatsoever in the context of Parkinson's for mild cognitive impairment. But what we definitely still see also in this independent cohort from the one that we did the original study on is that we still see these classic enrichments in the context of certain taxa in Parkinson's disease. And again, it's not just us who've seen this. This was nicely actually summarized here in a paper by Rosario, because they also focused on this enrichment in functions in the context of Parkinson's disease. So in Parkinson's disease, you see that there is an enrichment in the gene complement for mucin and host glycan degradative genes. And at the same time, you have a reduction in hydrolases that would be involved, for example, in starch or other polysaccharides hydrolysis. And so this was actually quite interesting for us in the context of Akkermansia in particular, because in a completely unrelated study that we did with Eric Martens quite some years ago, what we had seen is when we switched mice from a fiber-rich to a fiber-free diet, what we had seen is indeed that in the fiber-free diet, you get an expansion in Akkermansia mucinifila, along with other mucus foragers. So you basically take away one complex carbohydrate, and you then get an enrichment for organisms that are able to use host-derived complex carbohydrates. In this case, it is Akkermansia mucinifila in particular. And so the other aspect that is very important in this is that when you get this enrichment in Akkermansia mucinifila in the fiber-deprived diet, you basically have a thinning of the mucus layer because you have basically a lot of mucin hydrolysis going on. And so what is also clear is that this thinning of the mucus, in the paper we used a murine enteric pathogen and showed that the mice with a thinning or a thinned mucus layer, that they were more susceptible to infection by an enteric pathogen. And so that leads us to the hypothesis that if we see these enrichments in the context of Parkinson's disease in mucinolytic bacteria, that might actually increase their risk to exposure to toxins or, for example, amyloidogenic proteins that may trigger Parkinson's disease. And so this is then a potential trigger. This is from a paper by Sarkis Masmanian's lab at Caltech. And what they had shown is, in particular, that a microbiome-derived molecule, in this case, this is the amyloidogenic protein curly. So curly is basically expressed by Enterobacteriaceae, in particular Escherichia coli, for biofilm formation. So for example, for adherence to surfaces and then building up the biofilm matrix. And what Sarkis and his group showed, along with others actually, independent of their work, was that, so in particular CSGA, which is a subcomponent of curly, that it can act as the seed for the misfolding and aggregation of human alpha-synuclein. And so building on that work and our earlier work on fiber, we were interested to investigate whether higher levels of exposure to curly alongside a fiber-deprived diet, so when you have a thinning of the mucus layer, whether that would indeed lead to higher propensity for neurodegeneration in this context in a transgenic mouse line overexpressing human alpha-synuclein. And so what we did was basically we altered three factors, right? So diet, exposure to curly, and then also the genetic background. And what you see is, of course, is that you're already in a multifactorial space here. And so just to basically cut a long story short, what you see is if you basically have the mice on a fiber-deprived diet, that leads to the kind of microbial dysbiosis, one can call it, that we had already seen earlier, where you have then an enrichment in these mucinolytic bacteria. That then obviously has a negative impact on gut barrier function. You have a thinning of the mucus layer. And then if we add this insult of higher exposure levels to curly, so we use both a wild type overexpressing, or wild type, sorry, expressing curly strain, and we also had the knockout. And so with respect to the exposure to curly, you have an increase in aggregation of alpha-synuclein in enteric neurons. This then does spread to the central nervous system, where you also see degradation or degeneration rather. And this does have an effect on motor skills of these mice. So we did all of the classical assays here to look at potential motor deficit. And I have to say that there was a clear tendency in our data that the motor deficits were affected along the lines of what you would expect to see in Parkinson's, but that was not statistically significant, which is just probably a result of the fact that our cohort size was obviously not large enough, but all of the other effects that we saw were clearly statistically significant. The other thing which I would say is that if we had also left the mice probably live longer, we might have seen these more clearly. One would have to redo this study, first of all, with a larger cohort size, and then, of course, looking at motor deficits or motor skills longer over time. Okay, so that is what one can do in terms of distilling data down, high-resolution molecular data, to formulate clear hypotheses that one can then test, for example, in animal models. But another aspect I highlighted now, that all of this is very complex. And so I'll show you just now some so far unpublished data that give you some insights into the whole suite of omics and what that can teach us or tell us in relation to changes in the context of the gut microbiome, and how that then might tie into, in particular, Parkinson's disease. Okay, so I've shown you an iteration of this diagram before. Again, this is basically what we do is systematically generate metagenome, metatranscriptome, metaproteome, and metabolome data. We combine this with metadata, and then we have means of looking at differences across all of the omic levels. And this is from a more recent study where we had around 50 healthy control individuals, around 50 Parkinson's disease patients, and about 30 with a condition called idiopathic REM sleep behavior disorder. So that's a prodrome of Parkinson's disease. These individuals have a 90% risk of developing a Parkinson's condition in the subsequent 10 years. So they have a very elevated risk. Let's say, so we did this integrated multi-omic analysis, and then very similar to what I showed you earlier for type 1 diabetes. Again, we did not see any coherent, clear taxonomic differences, in particular in the metagenomic data, and we also did not see any clear functional differences in the metagenomic data. And so this is basically in each case here on the right, on the left and on the right, is a Permanova analysis where we have now all of the omic data, and then we can try to ascertain where we see the clearest discrimination diseased individuals. And so we can take this through for Parkinson's versus idiopathic REM sleep behavior disorder, control versus Parkinson, control versus idiopathic REM sleep behavior disorder. And so what you see here is actually that the functional gene expression resolved through the metatranscriptome, that is really the most discriminating omic data set. And then we can also start looking at confounders. So we know, for example, that constipation will have an effect on the microbiome, and we can see where in the context of the different omic data sets, we find a means of limiting, for example, that confounder. And also here you see that in relation to the functional gene expression resolved through the metatranscriptome, that you have the clearest discrimination in relation to diagnosis in that ohm. So that means that systematically generating this data is helpful, because at least it pinpoints us to where we may find the clearest discriminant signatures. Okay, so that is just summarized here. Again, the metatranscriptome of gene functions, along with the metabolome. You see that, for example, the metabolomic data here, you see differences, and you see that also here. So those are obviously the ones that one should focus on. And that makes also sense in relation to the metabolome, of course, because that most closely reflects the actual general community-wide phenotype. And it's obviously also least affected by confounders. So in the initial analysis, because it is the least affected by confounders, we focused really on the metabolomic data. And so here you see the list, in particular, of 11 identified features. So we measure, because we do untargeted metabolomics, so we measure a lot of unknowns. But here are the knowns, and here's the listing of the 11 most discriminant features that would discriminate between healthy individuals and those either with idiopathic REM sleep behavior disorder or Parkinson's disease. And then we can start looking at associations between these differences in those metabolites and then taxa. And so what you see here on the right-hand side is, again, according to statistical significance, certain genera of bacteria that are either enriched in Parkinson's disease or that you find at a higher level in healthy individuals. And what you can see here is that our known suspect from earlier is popping up Akkermansia, again as a genus. And then another taxon is Methanobravibacter. So Methanobravibacter is the genus of the most prevalent archaeon in the gut. And in particular, what we found was that the feature where we saw the clearest differences in terms of its levels is in beta-glutamate. So beta-glutamate is an isoform of glutamate, which is much less studied and hardly anything is known about it. And if you do a Google search, as I did initially, about beta-glutamate, what you find that was first reported as an osmolyte in archaea. But there's preciously little known about it. And here is just an overview. This is a diagram that I built to just give an overview of what we know about glutamate. So the most prevalent form, which is also the one that is relevant from a neurotransmission point of view, is L-glutamate. You have D-glutamate, and then you have beta-glutamate. And so now the question is, what is known? And so you see that we know most about L-glutamate. We know about 52 different pathways that feeds into. We know about 2 pathways that D-glutamate feeds into, but we know about 0 pathways that beta-glutamate feeds into. And what is also quite striking about this is that we see that beta-glutamate, so the levels of this are negatively correlated with genes that would be involved in L-glutamate, either metabolism or catabolism or anabolism. And so that is quite striking. And what we see at the same time is that there's a reduction in the expression of genes that have something to do with pathways that are feeding off glutamate. And in particular, what we saw was a reduction in glutamate-linked chemotaxis. So in particular, Chi-B, that is highlighted here, up here. And what you see here, in fact, is again, the levels of the individual metabolites that are statistically significantly different and their association with specific genes, both at the level of the metagenome and the metatranscriptome. And again, the clearest signal here we have is for beta-glutamate being negatively then correlated with genes that have, for example, a function in glutamate synthesis, such as, or L-glutamate synthesis, in particular glutamate synthesis. And that's not enough. So we see that chemotaxis is affected, and that is somewhat highlighted here again. We see chemotaxis that is dependent on glutamate being affected, and on from that is also flagella biosynthesis. So again, this we also see to be affected. So there's both a reduction in the context of Parkinson's disease of chemotaxis dependent on glutamate, along with flagella biosynthesis. And very, very stark, that is highlighted here on the right-hand side, what we see is that there is a marked reduction in the diversity of microbes that are able to express these two key functions that we've highlighted in the context of Parkinson's disease. So it means that there's somehow a kind of a bottleneck that individuals with Parkinson's, in particular their gut microbiome, goes through, which really reduces the diversity. And this is very complex, but this is just to give you an idea of the fact that we're able to resolve this data now to this level. And what you see is, in particular, this reduction in diversity in the context of Parkinson's disease. So these are basically genes that are both involved in chemotaxis and flagellar assembly, then flagellar assembly and chemotaxis. And so what you see is that for the most part, these functions are ensured by a number of different taxa, but then you really have specific signatures for either the individuals with Parkinson's disease or the healthy individuals. And again, in the context of Parkinson's disease in general, we see a reduction in the diversity of genes and the encoding and expressing taxa. Okay, so just to kind of conclude on this, so glutamate is super interesting because as you see in this community-wide network reconstruction, it basically feeds into very many different pathways. And so it is also important in relation to, in particular, chemotaxis and flagellar biosynthesis. And in particular, we now know that there are, in particular, also silent flagella, so that can in fact have an anti-inflammatory effect. So we're working on that with Ruth Lay at the moment. And obviously, the expression of those are dependent on the levels of glutamate, in particular L-glutamate versus beta-glutamate. And L-glutamate in particular is, of course, super relevant to look at in also the context of Parkinson's disease because it is, of course, the main excitatory neurotransmitter, and overactivity of it is, of course, known in the context of Parkinson's disease. Then on the side of the flagella, these are, of course, highly immunogenic, but as I highlighted already, they may also be silent, so they may, in fact, also have anti-inflammatory properties. And so what we're, of course, now investigating, because they trigger, of course, TLR5, whether those have a role in neuroinflammation. Okay, so how to investigate now such differences? So, of course, this data is very, very rich. And one doesn't have to be naive, right, to think about the fact that it's going to be very challenging to really test hypotheses. And so we're pursuing two approaches to test hypotheses. The first approach is basically that we're doing interventions. And one intervention that we're doing is therapeutic fasting. And therapeutic fasting has been proven to be efficacious in the context of microbiome-linked diseases, such as rheumatoid arthritis and Parkinson's disease. And so, for example, now in the context of an ongoing project, the expobiome project in particular, where basically fasting 30 individuals with rheumatoid arthritis and 30 with Parkinson's disease. And what this in essence is, in terms of the intervention, is a way of purging the microbiome from the gut. And then after the fast, the microbiome, of course, recovers. But what is important is that we're taking basically samples very tightly over time. And so we can see how the microbiome evolves and at the same time, how the host may respond. And so with that type of design, we at least are able to test some hypotheses because through the longitudinal dimension, we can, for example, through Granger causality, get really close to knowing whether a particular molecular factor or a combination of molecular factors may be linked to a given disease-linked phenotype. So that is one approach that we're taking. The other approach is that to do all of this in animal models, I've given you already the three-factor animal experiment that we did, and this already is proving to be difficult in relation to getting sufficient mice numbers together to get really to statistical significance at the level of the phenotype, in that case, the behavior. And so one has to have other models at least to do first pass experiments in. And one approach is to basically go to organs on chip. And that is also an approach that we've pursued now over a number of years. So we engineered over years now HUMIX, which stands for Human Microbial Co-Culture System. And with this, we're basically able to grow and sustain the growth of, in particular, strictly anaerobic bacteria, and that in very close contact with epithelial cells. And then what we've also more recently been able to do is to also integrate into it, for example, PBMCs and enteric neurons. And the other last point, other than, for example, also putting in linearized gut organoids now, what also we've been recently been able to show is that we can interface HUMIX with other organs on chips, in particular a liver on a chip. And we can do, for example, their drug toxicity testing. So, and those are a few highlights that I just wanted to highlight in relation to HUMIX. Some of you may know this system. So we've been able to put in induced pluripotent stem cell-derived enteric neurons. And the aim here is, of course, that we subsequently also put in enteric neurons, in particular, for example, from diseased individuals, most notably from Parkinson's disease. Then we have linearized organoids, the ability to grow those in the system. We've interfaced it with a liver on a chip. And with this, we were able, as a proof of concept, we were able to recapitulate the metabolism of a widely used anti-cancer drug, irinotecan, which is basically activated, in particular, in the gut microbiome, or by the gut microbiome, is then deactivated in the liver, but then reactivated again in the gut. And so we've been able to, with the interfacing of HUMIX with a liver on a chip, we've been able to recapitulate that. And then finally, one of the aspects that I already touched upon in the context of the fiber-rich versus fiber-deprived diet, a lot of this is happening, of course, at the gut barrier. And so one has to have means of testing the effect of, for example, specific bacteria on the gut barrier. And so for this, together with the group of Maria Tenge at Uppsala University, we've recently developed a new means of measuring a transepithelial electrical resistance based on impedance. Okay, so that brings me to the conclusion. So I hope to have given you some insights into the wet lab and dry lab methods that we've developed for integrated multi-omics of microbial communities, and how one can use that then to look through the different omic levels and see where you have discriminant features. I've introduced you to the concept of the expobiome as a complex trigger of disease processes, that you have functional disruptions in terms of the gut microbiome in chronic diseases. One that we've worked a lot on is type 1 diabetes, but then we see very similar effects in the context of Parkinson's disease. Generally speaking, that means that we should really consider function first and taxonomic differences subsequently. We obviously have a need for novel approaches to test hypotheses. For example, I talked about therapeutic fasting as an intervention that we can then track over time. We've also got HUMIX, which is a higher throughput system to do first pass hypothesis testing. And then I think the question that will occupy me until the end of my days at least is whether shifts in the microbiome are, of course, a cause or a consequence of disease. Okay, so with that, very many people, of course, to thank. And I'd like to thank you very much for your attention, and happy to discuss.
Fantastic talk. I loved it if I raised expectations high before we start a talk and then overfulfilled it. Wonderful. I start with a question. I have two questions. So the first question is, much you have shown us looks like a one-way road. So from gut, through the epithelium, from the bacteria through the epithelia, who then also working the other way around? Are the human cells producing something?
Yeah, for sure. I mean, the classic example there of is IgA, of course. IgA is obviously shaping the microbiome in a non-symbiotic way. And under specific disease conditions, the host may indeed shape the microbiome. So yes, it is definitely a two-way street, for sure. What we also like, for example, one host factor that we've looked at is, for example, stomach acidity. So I mentioned that a lot of what we see, some of the key taxa differences. So for example, in the context of type 1 diabetes, you see clear differences in different species of streptococci. Some are more pathobionts than others. And there we see that obviously stomach pH has a large role to play in whether you get transfer or not of those particular strains. So that's exactly, again, that it is a two-way street where really the host is shaping the microbiome. Finally, the one clear example that I also gave in the context of type 1 diabetes is, for example, the secretion of exocrine pancreatic enzymes, so in particular alpha amylases that are involved in starch hydrolysis. So if you have a difference in starch hydrolysis, that will, of course, also impact greatly the microbiome. So yes, it's absolutely a two-way street.
Other questions from the room? Christina?
I have a question to the metabolomics. So you probably did all of the metabolomics using LCMS-based?
And GCMS, actually.
Okay.
Yeah. And both targeted and non-targeted.
So,
yeah.
Because I was wondering, you showed mostly amino acids. It's one of the significant metabolites.
Yes.
But Bacteroidetes and Akkermansia are also known to produce a lot of like lipids, which are immunomodulating. So I was wondering if you grabbed those ones also in these studies.
So we didn't do the detailed lipidomic analysis in this first study. Now that we have, we have a couple of papers that are different levels of review at the moment. But we have, in the context of my expobiome project, so this is the ERC project, we are doing lipidomics and very, very broadly. So we should be able to pick up the differences, but I can't tell you yet whether there are really differences because we haven't analyzed the data.
Because for the glutamate, I was also thinking that these amino acids get easily acylated and converted and might also play a role in that one, like in the whole process.
Yeah, that's absolutely a valid point. Yeah. Agreed.
The second question I actually had was on the vesicles you mentioned, because you said that also many of them actually, the outer membrane proteins you pick up by the proteomic studies, that these are actually part of like mostly secreted proteins.
Yes.
And they are also mostly derived from Bacteroidetes.
Yes.
Is it true? So is it like that they are also really causing something, or it's just because they just use them as like to degrade material so that they can import?
If they cause something, I mean, this is just too early to say. I mean, because that data, again, was just from 4 healthy individuals. We are generating the same data. Actually, we just sent over to the US. We're doing this with Bob Hettig at Oak Ridge National Lab, the proteomics. We just sent them over the data on Friday to do all the database searches now. So it'll be interesting to see whether in that much larger study, we see similar signatures for the outer membrane associated, outer membrane vesicular associated.
Because they're probably also shed outside to do their actual functions and degrade polymers.
Right. Yeah, that's known, right? I mean, in particular, also for complex polysaccharide hydrolysis that they are decorated in essence with hydrolysis. Yeah, absolutely. Thanks.
More questions for Paul. I think you was first. Max.
I have a question concerning the Parkinson studies. So you mentioned a few bacteria, but I did not hear about Helicobacter pylori. Do you know anything about the relationship there, if there is one?
Yeah, so there is some, I mean, in the literature, you will find some links with H. pylori. We don't find any signature in our fecal samples. So there's a question whether, the fecal samples are really representative of the entire gut, right? I mean, this has been longly debated, and I think what one would need, and that is what we're working on now. So there's a company in the Bay Area actually that have developed this CAP-scan, basically capsule that you swallow. There was a paper in Nature earlier in the year. You might have seen David Relman is the senior author on that. And so we're looking at doing the same actually now in Parkinson's disease patients to get a proper transect. And if H. pylori is somehow involved, we would aim then to hopefully see that. Nevertheless, what I also wanted to say is that our multi-omic data is powerful in the context of also resolving gut topology, because we see that the ratios of metagenomic reads versus metatranscriptomic reads that we're able to map back onto metagenome assembled genomes, that there's differences at the levels according to where a given taxon would likely be the most abundant. So basically, given the different half-lives of, for example, RNA versus DNA, the RNA you typically find, right? And you are able to map many reads then from the metagenome onto a given taxon. Let's say, for example, active in the small intestine, but you will find relatively fewer metatranscriptomic reads. And so it's the opposite then for organisms that you find in large intestines. So we do get some topology that we can resolve, although I think that we need proper transects really to solve these questions that relate to the stomach, small intestine, and then the different compartments of the large intestine. Yeah, very relevant. Thank you.
Thanks a lot for the talk. I found it fascinating, especially how you go from correlation analysis down to molecular mechanism. In this curly example, that was really cool, but I think you had kind of a hint there that curly might be amyloidogenic. So I was wondering, how do you see this on a large scale?
Yeah.
You go down to like molecular mechanisms from the observations, from all the measurements that you do, and probably further, how would you convert that into some therapeutic approaches?
Okay. So this was actually what in the work that, I mean, Andreas just mentioned that I got back from Stanford, and that's actually the one project that I did there that was the most productive. So I basically went back and did stuff myself again for a very long time. It included building a bioinformatic pipeline, and that was specifically for identifying amyloidogenic proteins. And I found very, very many, in particular, small proteins that are amyloidogenic. And so out of those, I mean, just going down the molecular mechanism in the context of basically amyloid formation on the human side. And so actually of the 10 initial candidates that I got, 4 of those are amyloidogenic, and they can indeed cross-seed amyloid beta aggregation. So that is one thing that one can get there. And one important development there is AlphaFold 3, for example, looking at really intramolecular interactions. So I think those approaches are super powerful, and they will be super powerful going forward. So you have to get to the point of having narrowed it down in terms of the molecular space, because this is one thing that I also learned from my time in the US, is you have to conceptually think about this as a funnel. How can I build a funnel that gets narrower and narrower, such that I get to one molecular class that is compelling to look at from an experimental point of view, because it is very complex. And I don't know how we will go from having narrowed it down again back up. That will be a challenge for the future, but I think there's super exciting developments, at least in relation to protein structure prediction and interaction. Fabian.
One question to your cross-sectional and longitudinal Parkinson's cohort that you want to use to try different hypotheses. How do you consider the fact that many of these Parkinson patients probably have an oral LDOPA treatment?
But these were all naive. So these are newly diagnosed Parkinson's disease. I didn't mention that, my apologies. Yeah. But they have not been treated at all.
And have you run any studies where you try to find what's the difference in these LDOPA treated patients, for example?
We have not. And the reason is that we want to understand potential pathogenic mechanisms at play. And I think that if you look at people that have been treated, I mean, also we know that L-DOPA is also massively metabolized by the gut microbiome. That may actually be similar to metformin. Metformin is a diabetes drug, has also been shown to massively impact the microbiome. So I think you have to think whether you want to really look at cohorts of individuals that are undergoing treatments where the treatments probably will have already a marked effect on the microbiome. I think you will not get to any point in causation, not least because probably even looking at the gut microbiome of individuals that have a manifest diagnosis is probably also too late. You would probably want to look at their gut microbiome 20 years earlier. But yeah, so we have to work with what we have. But I think from a general point of view, I think treatment is something that one should probably exclude as much as possible.
Alex.
Thank you for the talk. You mentioned that you're also interested in oral microbiome, and since the whole talk was mostly about gut microbiome, are you somehow, or in some studies, compares oral and gut?
Yeah, exactly. Yeah. So that was our segue. Actually, we wanted to understand how the oral microbiome may be a reflection of environmental exposure and how the oral microbiome may then affect the gut microbiome. And initially, we focused on type 1 diabetes, but we also looked at colorectal cancer, because in colorectal cancer, you might know that there is a clear enrichment in facultative oral pathogens, Fusobacterium nucleatum, Streptococcus stomatis, and so on and so forth. And so we were interested in seeing whether we could find differences already in the oral microbiome. And for colorectal cancer, that is indeed the case. For type 1 diabetes, that is the case in relation to specific strains of streptococci. And what the transfer depends on is, for example, stomach pH, yeah. Which maybe people say, well, that's not news. Yeah, but it's important to show it, though, I think. Yeah.
Perfect. Okay. Let's thank Paul again. And there are some Berliners outside for frapping.